first, just following the stuff about breathing and respiratory capacities, the book then gets into how we control breathing. And I want to go ahead and mention this now. I'm not going to talk about it a whole lot. After we cover the gas exchange stuff, then I want to talk a little bit about what leads to regulation to some degree. But looking at this material here, breathing is controlled by a couple or few centers located in the brainstem. The primary center that controls breathing is in the medulla, and it's called the dorsal respiratory group. Neurons in this location send axons out that go down through the rest of the brainstem and into the spinal cord and cause contractions of the diaphragm and external intercostal muscles. Those are the basic muscles of breathing. They're responsible for just normal, everyday breathing, not any speech production kind of breath control or anything like that, just basic breathing. Actually, I want to show you one thing that doesn't really work on the capture, but this model up here shows the relationship of the spinal cord to spinal nerves and that sort of thing. Coming out of the cervical spinal cord, between cervical level 3 and 5, are axons from the dorsal respiratory group. That's not represented in the picture in the book, but it is true. And they end up going into this nerve right here, which there, of course, be one on the other side, but it's only represented on one side of this model. This is the phrenic nerve. The word phrenic refers to the diaphragm. And so this is the nerve that actually goes to the diaphragm. Now, the diaphragm is going to be located further down in the mid to lower thoracic region. So it's a little strange that the nerve actually leaves from the cervical level to go down there. But there's a very important reason for that, which has to do with the fact that this dorsal respiratory group nucleus here is autorhythmic. Now, where else have we talked about autorhythmic cells? Yeah, in the heart. So the SA node, AV node, those sorts of clusters of cells in the heart are autorhythmic. They generate an action potential, and then they slowly depolarize back to threshold before they start another one. The rate at which that happens sets up the rate of the heartbeat. The reason why those are autorhythmic is because there's a sodium leak channel in the membrane that slowly depolarizes the cell between action potentials. The same thing is happening in the dorsal respiratory group. Now, a big difference between those two things is the SA node and the other parts of the heart that are autorhythmic. Those are cardiac muscle cells. Those are conductive cardiac muscle cells, but still, they're cardiac muscle cells. This is a nucleus in the brainstem. Okay, so these are neurons. These cells will also slowly depolarize until they reach threshold and fire off. The rate at which they do that, sort of the basal rate, is about once every 5 seconds. If those are firing once every 5 seconds, causing contraction of the diaphragm and external intercostals, what respiratory rate does that mean that it's setting up? How often are you breathing if you're breathing once every 5 seconds? Actually, compared to the heart rate, not that often. Okay. Every 5 seconds is 12 breaths per minute, okay? As opposed to the heart rate, which would be about 100 beats per minute left to its own devices, and it's actually slowed down by the parasympathetic system to about 72 or so. Okay. But those are action potentials that are firing for resting heart rate about 72 beats per minute once every 0.8 of a second. Okay. This is one firing every 5 seconds. And that's sort of the standard respiratory rate that we usually think of here. But respiratory rate actually can vary quite a bit, between about 12 and maybe 20 breaths per minute. So if it's 20 breaths per minute, how often is that? Are those neurons firing? Every 3 seconds, right? Okay. See the relationship there? Every 5 seconds, there's 12 5-second periods in a minute. Every 3 seconds, there's 20 3-second periods in a minute. Okay, that's math, folks. Sorry, I'm making you think like that here, but so that's what the dorsal respiratory group does. Okay, it will send information over, or send the action potentials over to the ventral respiratory group, which can control the internal intercostals and the abdominal muscles, which are the muscles of inspiration, as well as what are not really, well, some of them are kind of represented here. I think these are the serratus muscles, and then this is the pectoralis major, which obscures most of the pectoralis minor, which is down in here. But those are other accessory respiratory muscles. Okay? And when you need to take deep breaths or you need to force air out, those muscles controlled by the VRG will lend their work to that. Back to the phrenic nerve, these fibers that are coming out are going down to these various muscles. The ones that are going to the diaphragm actually come out of the spinal cord in the 5th to 3rd cervical region and extend to the diaphragm as a nerve in the periphery, as opposed to going down the spinal cord to the thoracic level, 8 or whatever it might be, and connecting directly to the diaphragm. Why is it advantageous for those fibers to be peripheral for most of their path to the diaphragm as opposed to central? It's a difference between axons in the central nervous system versus the peripheral nervous system. No. The speed of conduction is based on myelination and fiber diameter, and those couldn't be comparable in the 2 locations. What's really different between axons in the central nervous system versus axons in the peripheral nervous system? Sorry? What types of glial cells do we find insulating central axons versus insulating peripheral axons? You remember AMP1?
All of the dangerous things.
That is one of them. Which one is it?
You tell me.
Well, you're halfway there. You can finish that thought.
The central?
Central. Thank
That's
you.
what I mean.
That's what you, yeah. What do we call the glial cells that insulate peripheral axons? Not oligodendrocytes. Schwann cells, right. Okay. Now, there's a fundamental difference in what those 2 cells do for the axons. Okay. I'm going to take a long shot and see if anybody knows what that difference is.
Between the Schwann and the oligodendrocyte.
Right. They both insulate fibers the same way. They're myelin. But there's another feature that one of them has that the other one doesn't. Anybody know what it is? I mention it briefly in AMP1. I don't know if my colleagues do necessarily. It has to do with injury.
They can read.
Read what?
I can't think of 8 o'clock at night. They can regenerate.
Or regrow would have been an acceptable word there. The glial cells don't do the regrowing. The axons do. But the glial cells control when that happens. Which ones allow axons to regrow? Yeah, the peripheral ones. Central ones actively inhibit axon growth, or not inhibit it necessarily, but slow it down. There is some evidence that axons in the central nervous system, if you break your neck, will regrow, but it takes decades to see any real significant change. Okay. There's a lot of research trying to speed that up and find ways to do that. But in the periphery, if a nerve is damaged, the Schwann cells actually help the axon regrow over the damaged site. Okay. So back to the phrenic nerve. We have these axons leaving the spinal cord very quickly after they come out of the brain, and they're in the periphery to go down to the diaphragm. Why would that be advantageous? Right. If they're going through the spinal cord and you have a spinal cord injury in the upper thoracic region, you're going to sever any nerves there. And if those are controlling breathing, then you're going to lose the ability to breathe. Okay. If the nerves are in the periphery, which they are, irreparable damage is only going to occur when there's a break in the neck above about C5. Okay. Or actually, if there's a break between C3 and C5, some function can be reserved. But above that, above C3, then you'll definitely sever those nerves and you'll lose the ability to breathe.
So is it like kind of like an automatic protection?
I don't know if I'd use the word automatic, but it's an advantageous adaptation, I suppose. You could just think of it this way. Organisms that had phrenic nerve fibers that were central until they came out at the level of the diaphragm, if they suffered some sort of spinal cord injury, would die and not reproduce. And then those that had the fibers in the periphery were more likely to survive spinal cord injuries, and therefore be able to pass the trait on to offspring. Now, the other structures here, which I'm not going to talk about a lot, but I want to mention them. In the pons, these pontine respiratory group nuclei, called the pneumotaxic and the apneustic center, have an influence over the dorsal respiratory group. Dorsal respiratory group is autorhythmic, so it can work on its own to keep you breathing. But higher centers in the pons can influence what's going on there. And the book describes those two centers in more detail. Also, what's not really pointed out in this picture, because the insert is really just of the brainstem, but the cortex can send information down to the pons, which will influence the medullary centers and regulate breathing a little bit. If you're going to speak, of course, that's a completely conscious, voluntary response. So you're going to have cortical speech centers sending signals down into this to help control breathing so that you can produce speech sounds, that kind of thing. There's also input from the hypothalamus, which is located right here in the picture, which if you're frightened or anxious or something like that, then your respiration is going to change. You'll, likely hyperventilate or something like that. And that can have control from those levels there. So again, the book kind of gets into this next after talking about breathing. So I wanted to mention it there. And there's some talk about things that play a role in regulating breathing rate and depth of respiration. And that's the end of this section. I want to come back to that in a little while, because what's more important is to talk about the gas exchange and transport of gases part of things. And from that, we'll understand a little bit more about what we regulate in breathing. So gas exchange is essentially the two things that I had, that I still have up here: external respiration and internal respiration. And in the book, that's gas exchange is one section. The next section is transport of gases, which is what's going on in the blood. And we'll look at that in a second. Now, to understand what's going on with gas exchange, we have to understand gas a little bit more in depth. We need to understand a couple of other gas laws. One of those is Dalton's law. And actually, let me pull up. I have a link to a video I want to show you real quick in Prezi. Oh, before I get into that link, while I'm thinking of it, this picture here shows the bronchial tree. The trachea and the primary and secondary bronchi have been injected with a clear plastic, and then the tertiary or segmental bronchi have been injected with colored plastic. And so as you look at this, you can see how the bronchial tree, as it branches, stays segregated. This branch here that's colored in white is one segment, and this blue one here, and this red one, the orange behind it, the yellow, the light blue, brown or green or whatever color that is, and this color up here. Those are different regions of the lung. If something blocks the bronchi at this level, only the white stuff is affected. All of the other segments of the lung function perfectly normal. Okay. And if something's blocking this light blue one down here, then this segment is affected, but none of the other surrounding parts are. If something's blocking the bronchus that's going to this inferior lobe, then that lobe will be affected, and the superior lobe is fine. Okay. Having an entire lobe compromised is usually more problematic than just a segment. But so there's a little bit of segregation in this so that we can have a little bit of control over things. The model I was showing you earlier is kind of old, and it's got colored branching patterns showing where these things start, but it really doesn't expand as well as this plastinated picture shows us. Okay. Anyways, so while I have that, I just want to show you. But down here are some things to mention about gas laws. So I was talking about the volume-pressure relationship earlier. So if this is volume of gas, if the volume gets smaller, the pressure gets higher. And if the volume gets larger, the pressure drops. The way it's represented here, this particle is moving around within this space, just like an oxygen molecule or a nitrogen molecule is. And it runs into the wall of the container as it's moving around. The smaller the container, the more impact there will be. The larger the container, the fewer impacts. Each impact is a little bit of force being exerted. That one impact isn't a whole lot of force, but as all of the different particles in the air are all moving around and all running into the surface of the container, that together adds up to the pressure in that volume of air. So if the volume gets smaller, there's more chances of the things to impact the walls. And if the volume gets bigger, there's less impacts, and so less force exerted. That's kind of where the pressure-volume relationship comes from, which I talked about before. The next thing I want to talk about is Dalton's Law, which is sort of stated here, but this video does a pretty good job of explaining it. Now, this video is from an outfit called Mind Bites. It's a YouTube video. And it's actually just a fragment of a larger lesson. If you would like to know more about this lesson, you can, I don't know, subscribe to or pay for access to Mind Bites or whatever. I know, but this is a free little segment, and it's just about Dalton's Law. Now, it's a little bit odd of a video. As you can see from the colored balloons behind them, they're trying to make it look kind of, I don't know, fun or exciting or something like that. And there's some sound effects that kind of go with that, almost making it seem like it's pitched at kids, but the gas law information that he's dealing with is a little bit advanced for kids. So it's kind of a weird mix. And there's a point where he probably is not hearing the sound effects that they add in later on, but the expression on his face just strikes me as, oh my God, why is this happening around me? So let me just play this out. Oh, shoot.
What I have here is a balloon filled with ordinary air. Air is a mixture of gases, and you probably have thought that air was mostly oxygen. After all, oxygen is the piece that you're most interested in. You need it to survive. In fact, air is mostly nitrogen, N2. It's about 78% nitrogen and only 21% oxygen. What we're going to be talking about in this section are mixtures of gases. And the question that we're going to consider here is, we have a balloon. There's pressure from the gas on the inside of the balloon exerted against the outside of the balloon. If it's a mixture of nitrogen and oxygen, how are the forces of the individual gases related to pressure, and how are they related to the total pressure of the balloon? So in other words, each of the individual gases presumably exerts some pressure, and how is the component of each of the individual gases related to the total pressure of the balloon? And get rid of this. To answer that question, what we need to do is define a quantity first called the partial pressure. Okay. So here we have a mixture of gases. It's inside a container of volume V, and each of the individual gases is indicated by different dots. So we've got some red dots and we've got some blue dots. Now, what I want you to think about is, suppose we took everything out of the box except for the red dots. So we have a quantity of gas A in the volume V, and we can measure with this pressure gauge the pressure due to A just in the box all by itself. And we're going to call that quantity the partial pressure of A. So again, piece of A, the partial pressure of A, is equal to the pressure due to A as though it were alone inside the container. Okay? This is in the context, again, of a mixture of gases. Well, we can do the same for B. B, let's say, are the blue dots. We can have just B inside the box. We can define piece of B as the pressure due to B alone in the container. And now we get to Dalton's Law. John Dalton, in the early 1800s, discovered that the total pressure exerted by a mixture of gases equals the sum of the pressure of the component gases. And this is now known as Dalton's Law of partial pressure. If we look at an algebraic representation of Dalton's Law, it says that the total pressure inside the container is equal to the partial pressure of A plus the partial pressure of B plus the partial pressure of C plus whatever else might happen to be in the container. And the really important point of this is that basically what this says is that A is oblivious to the presence of B and C. And similarly, B doesn't know that A and C are in there. So the gases don't even interact at all. And that is a tenet of the ideal gas idea, that the particles are so far apart that they're not really even interacting with each other. And so it really doesn't much matter whether the gas.
Okay, so he goes on a little bit there, but that gets to the basic idea of what Dalton's Law is about. Okay. So having shown you that, let me go to the book here. Okay. And this table here and the figure below it, basically it addresses the same thing that the video just showed. So down below we have a representation of what he had in that drawing. So here's a mixture of two gases, which are specifically named oxygen and nitrogen here, but that could be the red dots and the blue dots that were in the picture you just saw. If we removed one color dot from it, then we have the same volume, but with just that particular gas in it, and we're just going to see the pressure related to that one thing. Or we could do the opposite and remove the other color. We just see the pressure related to that. Okay, that's what he was showing in that little drawing there. This table is showing the partial pressures for the components of air based on the total pressure that we see at sea level. So earlier when I was talking about pressure and volume, I said 1 atmosphere is equal to 760 millimeters of mercury pressure. Okay, so that's our total pressure there. Nitrogen makes up about 78.6% of the air that we breathe. It actually does nothing for us. We breathe it in, we breathe it out. It doesn't do anything. It really just takes up space. The reason for that is that oxygen is the really important thing in the air that we're breathing, but we don't want too much oxygen. Okay? Too much oxygen leads to oxidative reactions, which if your environment is overly oxidative, you're going to have fires and explosions left and right. Okay, so we need to limit the amount of oxygen in the air around us so that we don't, all spontaneously combust. So that's what the nitrogen's there for. It just takes up space in the air. The nitrogen's partial pressure is 597.4 millimeters mercury. We don't actually measure that separately. All we have to do to get that number is multiply 78.6% by 760, and that's what we get there. Likewise, oxygen, which is just shy of 21%, if we multiply 21% by 760, we get 159 millimeters of mercury. Water vapor in the air is about 4/100%, which gives us 3 millimeters of mercury. Carbon dioxide is about 4/1000%, which gives us 0.3 millimeter of mercury. And then the other gases that we find there, like carbon monoxide and other sorts of gases that are present. I guess there's argon and those sorts of things in very, very small amounts. They, as a group, represent 6/10,000%. Now, this has to be a typo because 6/10,000% times 760. I see what they did. They didn't put the decimal point in the right place. So yeah, that should be 0.005 millimeters of mercury, which is more in keeping with the numbers there. Very simple mistake, and actually when I was trying to figure out myself, I made the same mistake. If you multiply 760 by 0.0006, you get 0.5. If you multiply 760 by 0.000006, which is actually how you multiply it by 6/10,000%, then you get the proper value here. So they just did the exact same thing I was doing. Too many zeros and decimal points, you get kind of thrown off. But that really should be 0.005 millimeters of mercury worth of pressure, just so it all works out. But all of those add up to 760 millimeters of mercury, your total pressure. And all of these values add up to 100%, roughly. There's some rounding errors in there, I'm sure, but that's basically how it works out. So that's what the Dalton's law is getting at. 760 total pressure is equal to the partial pressure of nitrogen plus the partial pressure of oxygen plus the partial pressure of water vapor plus the partial pressure of carbon dioxide plus everything else. Okay. And we can actually figure these precise partial pressures out just by looking at the percentage composition and the total pressure. What's suggested down here is nice as a thought experiment, but you couldn't actually remove just one gas from a container. So like if in this room, if it was closed off, we could suck everything but the oxygen out. One, we'd probably all die. But two, there's no way to only take certain gases out of a mixture. Just not possible. So it's easier to use something like mass spectrometry or chromatography, whatever, that other thing that's not coming out of my mouth this late at night, to get the percentage composition, and then just multiply those percentages by the total pressure, and you'll get the partial pressures. You are not going to need to figure out partial pressures because we know what they are in all the different circumstances where they apply. But it's important that you understand where they come from. Okay. The other thing to consider is Henry's law, which is the solubility of gases in liquid. And so going back to the presentation, I don't have another video to show you, but rather, sorry. Nope, not what I want to do. There we go. What Henry's law is saying is that whatever the partial pressure of a gas is in an air phase determines what it is in a adjacent liquid phase. Really what it means is that gases diffuse into liquids easily. We can imagine the gases in this room are all mixed in just a homogeneous concentrations within here. Okay? Any little part of this room that we sample is going to have those gases in the same proportion. They'll just diffuse to equilibrium throughout the entire room. The same's true of gases in liquid. They'll just diffuse to equilibrium within that liquid. And in fact, the same is true when there's a gas phase and a liquid phase up next to each other. The gas particles, carbon dioxide, oxygen, whatever it is, in the gas will diffuse into the liquid as if it's the same medium as air. There's nothing keeping it from moving back and forth there. So to kind of illustrate the idea of what Henry's law is about, these beakers with plungers on them are kind of showing you what happens as we alter air pressure and how that affects what's going on in the liquid. So if we start with a beaker that's filled with this much water and air, which would, let's say, are equal volume, and we push the plunger down so that the air phase volume decreases, the pressure in the air is going to increase. That's a simple pressure-volume relationship we were talking about before. Now, when there's a higher pressure in the air phase, the particles in the air are going to fuse into the liquid until they reach equilibrium. And just as an example of what we're really kind of talking about here, that's what happens in a soda can. Okay. So at the bottling company, when they're making Coke or whatever, they put the Coke mix in there, and then they inject carbon dioxide into the can and seal the can so that the pressure in here is higher than what's outside. The partial pressure of carbon dioxide in the air and the liquid phase here is going to get to equilibrium. And there's going to be a pretty high amount of carbon dioxide in there. If we suddenly remove the lid from our beaker, the gases in the air phase will come to equilibrium with the rest of the air next to it pretty much instantaneously. And so the gas that's in the liquid phase will move out to diffuse to equilibrium. until the partial pressure of air of the gas in the air phase is equal to the partial pressure of the gas in liquid phase, which is what happens when you open a soda can. Okay. Now, this isn't instantaneous between the liquid and the gas phase, liquid and the air phase, which you've experienced with soda. When you open up the can, the bubbles that you feel, the fizziness, is really because the high partial pressure of carbon dioxide in the soda is diffusing out. Okay. Once it reaches equilibrium, that's when the soda is flat. Okay. When the partial pressure of carbon dioxide in the soda is equal to the partial pressure of carbon dioxide in the air around us. And we just saw the partial pressure of carbon dioxide in the air around us is 0.3 of a millimeter of mercury of pressure, which is very low. I have no idea what pressure the carbon dioxide is in the can before it's been opened, but there's a pretty big differential in air there, in pressure there. And it takes a while for the carbon dioxide to come out of solution and we get to the flat stage. Okay. So that's what Henry's law is about. Okay. Do people need me to turn the lights back on to see things? Everybody okay with the very low light level we have here? Is everybody awake? Okay. Not turning the lights on will actually kind of help psychologically. It'll feel cooler in here, even though it might not be. So I'm going to leave the lights out. I think there's enough light from the projector and various computer screens around the room that people can kind of see what's going on. But if you have a problem seeing things and trying to take notes or whatever, let me know and I'll turn some of the lights back on. So that's Henry's law, which the book is talking about here, about the solubility of gases in liquid. Basically what it says is gases dissolve into liquid just fine, and we treat them as if they're in air. We talk about the partial pressure of gases in a liquid rather than the concentration. Okay. It's just the way that we describe that. Now here's another table about partial pressures. This is the composition and partial pressure of gas in the alveoli. When you breathe air in, there's going to be exchange of gases, which we'll talk about. Oh, we're not going to get to talking about, apparently. But let me talk about this for a second and wrap up here. The amount of nitrogen, percentage-wise, changes. And really what's happening is the nitrogen's not changing, everything else is. So it ends up being less of a percentage of the air composition there. Oxygen goes down also, and that's because a lot of the oxygen is moving into the blood. So the percentage composition drops rapidly. Water and carbon dioxide are higher. The water is higher because, as I was talking about earlier, when you're breathing in, that sound is the air moving around in your nasal cavity where the air is being hydrated. So the water vapor content goes up quite a bit. And then the carbon dioxide is higher because there's carbon dioxide coming out of your blood very rapidly. So the percent composition changes, which means that the partial pressures are different also. The ones that we really want to pay attention to is the partial pressure of oxygen is 104, just over 100. And the partial pressure of carbon dioxide is 47, a little less than 50, a little more than 45, thereabouts. As air is exchanging, as these gases are exchanging between the air in our lungs and our blood, these pressures are really going to define how much of those gases are in the blood. Okay. And so for external respiration, what we see is an exchange of oxygen from the air in the alveolus into the blood, and then carbon dioxide from the blood into the alveolus. The partial pressure of oxygen is higher in the alveolus than in the blood. So it's going to move into the blood. The partial pressure of carbon dioxide is higher in the blood than in the alveolus. So the carbon dioxide is going to move out. They work independently. And in that little video I showed you there about Dalton's law, he mentioned briefly what's called the ideal gas concept, or whatever term you use there, which is, it's not exactly the way that gases work, but we always treat them as working independently. Within the air in this room, there's oxygen molecules and carbon dioxide molecules and nitrogen molecules all whizzing around back and forth. The distance between any two molecules is so great that the chances of them actually running into each other is effectively zero. And so ideally, we consider gases as working independently within a mixture. So oxygen will move in one direction and carbon dioxide will move in the opposite direction because they don't really have an effect on each other. They don't interact with each other. They're just following their own partial pressures back and forth. So I didn't intend to not finish this up tonight. So we'll pick up with this next time just to finish this thought, and then we'll get into the digestive system next week. So have a nice night.